Hello, welcome to Virtual Medical Tutors. Today we're diving into a fundamental concept in medicine, the central dogma of life, first proposed by Mr. Francis Crick in 1958. The dogma here simply means a principle or belief that cannot be doubted. This is a theory stating that genetic information flows only in one direction, from DNA to RNA to protein, or RNA to directly to protein. There are, however, a few exceptions which I'll talk about at the end of this video. This principle, as basic as it looks, forms the backbone of how genetic information is expressed within our cells and is essential to understanding everything from basic biology to advanced medicine. The central dogma of life explains how genetic information flows from DNA to RNA through a process called transcription to protein through a process called translation. I'll talk about these processes in better details in my next video. You can liken this whole process to a bakery or a big restaurant. Think of the DNA as a cookbook that contains all the recipes needed to make a variety of dishes. This cookbook is stored in the kitchen's main library or archive, which is the nucleus. For transcription to happen, just like a chef might copy a specific recipe from the cookbook onto a note, as the mRNA in this case, to use in the kitchen, the cell transcribes a specific gene from DNA into mRNA. This mRNA is a portable copy of the recipe that can be used directly for cooking. Now the mRNA, as the recipe note, is taken to the kitchen, the ribosomes, where it's followed by the chefs, the tRNAs, to prepare the dish. The ingredients, that's the amino acids in this case, are brought together by the chefs according to the instructions in the mRNA to create the final meal, the polypeptides, which is then modified to form proteins. These proteins go on to perform critical functions in our bodies, from building tissues to regulating enzymes. As I said earlier in this video, there are some exceptions to this sequence. While the central dogma generally holds true, there are exceptions. For instance, in some viruses like HIV, the process can go in reverse. This is called reverse transcription, where RNA is used to create DNA. As you can see in this diagram, RNA becomes DNA so that it can integrate with the human DNA to be transcribed into multiple copies of RNA, and hence how it replicates in the body. Another example is that of prions. Prions are infectious proteins which replicate without going through DNA or RNA intermediates. These exceptions highlight the complexity and adaptability of life's processes. Prions are responsible for the rare disease Creutzfeldt-Jakob disease. It is a disease that causes degeneration of the nervous system. So how does this theory benefit the society? Understanding the central dogma is critical in medicine and biotechnology. For example, in gene therapy, scientists aim to correct faulty genes by ensuring the right proteins are produced. It can help prevent cancer, diabetes, hemophilia, and a host of other diseases. Similarly, many drugs work by targeting specific stages of transcription or translation to treat diseases. To sum up, the central dogma of life basically describes the essential flow of genetic information from DNA to RNA to protein. This process is foundational to life itself. If you found this video helpful, don't forget to like, subscribe, and share it with others. If you have questions or topics you'd like us to cover, drop a comment below. Thanks for watching, and see you in the next video.